Hi everyone, thank you so much for joining today's webinar. Today's webinar, I'm going to be talking on M13 primers, basically, which can be used for sequencing purposes, which means for Sanger sequencing purposes. My title of my presentation will be Tale of Tales with M13 primer for COVID-19. For today, what will I be covering? So I'm going to give some introduction, what is Tale of Tales means, and then what is M13 Tail Primer, what is the workflow, especially the workflow from our facility, our lab, and then what are you ought to expect, especially results maybe on COVID-19 samples. How do you prepare your samples and how do you submit them to us? And finally, I will conclude. Now, let me start with the introduction. Okay, so in the beginning, basically PCR amplification, maybe using any DNA polymerase. So it was actually used as an selective enrichment. And later on, as technology develops, we have the ones in sample amplification. So it was soon adapted for multiple application. Now, to facilitate downstream application means after PCR, we want to look into the downstream application. So non-specific utility sequence, they are actually added at 5 terminals of primer, which means 5' at the beginning of the primer. Now, the tails that are added or the sequences that are added, actually it is for few purposes. Now, the first purpose definitely is to for cloning purposes. So you have the RE sites, and then we also insert some universal primers for sequencing purposes, for especially for Sanger sequencing. And there's also some RNase promoter sequences for in vitro transcription. So these are some of the sequences or basically tails that we add to facilitate the downstream application. Now, basically, the general function of why we add these particular tails is basically to improve the robustness and overall performance of a diagnostic assays. So again, main aim, it is to improve disease and also pathogen detection by enhancing the amplification process. So this is the basically why we add the tails actually. Now, um, why do we add tails? And if we don't add, what are some of the problems that we actually encounter? Now, we know that some of these microorganisms, especially virus, they tend to have a very high divergent targets. So basically, they tend to lack a universal conserved region. And this is very true when it comes to viruses, because viruses, they tend to have a higher mutation rate, right? So they tend to have higher divergent targets. And then degenerate bases and incorporated bases are captured at the most observed genetic variation. So because when we tend to align, we want to get maybe a conserved region site that we can cover the whole overall genetic sites of these particular genes that we want. So we tend to put in some degenerated bases. Now, what are the problems we tend to have when we have degenerate bases to amplify this region is we tend to have problems at primer binding sites. Okay, now though our primers are, for example, because primers, it has to be very specific. So when we do not have a primer that is specific, so when we want to sequence, we tend to have problems in the binding site. Not only that, sometimes it also results in mutation, indels, all right, and also degenerate bases. So all these tend to bring problems to whichever sequences that we want to analyze downstream. And then there's also some mismatches like between primer and target sequences, thus giving a serious impact on the amplification efficiency. So mismatch in the sense that maybe we tend to see a multiple bands instead of seeing a very, a single intact band. But when we do, when we don't have these kind, of tails, and when we have a shorter sequence reads, we tend to see more mismatches between the primer and the target sequence. And then what's more, when especially when we tend to have the template concentration, which is literally low, or if not, there is actually inhibitors which are present. Now, this is very true when we have diagnostic samples, especially now with the recent outbreak of COVID-19, right? So we tend to have multiple samples. Okay, we want to see whether a patient is actually positive COVID or negative COVID. So we take a swab sample. So now, literally, if the patient is not showing any symptom, all right, maybe the concentration might be low. Okay, that is one. And another one more, even if they are showing, we tend to have some inhibitors, like maybe some uh, the samples are basically not clean enough for RNA extraction and things. So when we have all these, it also tend to bring to the downstream part, whereby we tend to have problems in analyzing them to verify them. Okay, so this is very true when it comes to diagnostic samples. So this is what some of the problems that we have in general when we do sequencing or amplification, especially when it comes to diagnostic samples. Now, how tailing helps? Now, basically, when we have tails, basically it tend to improve the robustness amplification in PCR. Okay, so you may have your qPCR product, but when you have some samples like this, we tend to have better robustness, better improvement. Now, it also improves the overall performances um, of the amplification process. It also can neutralize the destabilizing effect of mismatches, because when your samples are literally or your templates are literally shorter, now you can actually by using these particular tails, it tend to stabilize. Okay, and also when we use primer tailings, generally, so primers you can use actually for you can use more primers. Okay, we can have variants that you can actually participate in multiple PCR reaction. Not only that, tailing, like what I said, it increases the binding affinity, because now, despite of having primers with degenerate bases, now you do not need to sequence with that. So it basically uses the tailing sequences to sequence, so it increases the binding affinity. So when it increases the binding affinity, it reduces the error. Okay, so let's move on to M13 Tail Primer. So this particular tail primer is in aspect for Sanger sequencing, which means it is more towards sequencing aspect. Now, what are the limitations that we see when it comes to diagnostic samples, especially for COVID-19 samples? Now, screening and detection for COVID-19 samples actually utilizes qPCR fragments. So basically, they are to screen whether they are positive or they are negative. So basically, we do a qPCR fragment. So most of these particular target genes, for example, we have RDRP, we have E gene, N1, N2 gene. So all these particular fragments are much more shorter, whereby they are less than 100 base pair. And then also these particular viruses, like what I've explained earlier, they tend to have high variability regions. So to obtain full coverage, all right, full coverage, you have to generate a degenerate primers. Okay, and then these primers, they are only suitable for amplification. So when we want to sequence, we would see, like what I've explained earlier, you will see primer binding problem. Sometimes you tend to have mutation. So all these particular sites comes in. So definitely you need to have a very specific binding. So how M13 tend to help here? So M13, what happens is, when we use M13 for Sanger sequencing, it's able to tabulate regional mutation. Basically, you can use these particular primers where you can amplify. And if you want to study regional mutation, especially you want to screen for diseases. For example, say in RDRP, maybe you have few patients, and then you are interested to know if there's any mutation in this particular site or any genes of your choice. You can actually, but it's a qPCR or perhaps maybe shorter fragments. You can always use these M13 tail primers. And this M13 tail primer, basically it's easy to use and it has very simplified workflow, which I will later share to you. So basically from sample, from the time you submit your qPCR sample until you get to know what's the mutation sites and stuff like that, the workflow actually can be completed in 5 days. So this is much more faster compared to cloning, because if you want to know that usually for qPCR fragments that is lesser than 100 base pair, usually we tend to have problem in sequencing. So the alternative way what we have usually we will do is we tend to clone them into a cloning vector, transform it, and then you have to lyse the particular E. coli strain, and then you get a purified plasmid, and then you sequence. So literally, this one may take up to about close to 2 weeks. So what we are doing now here is we can cut short that, and we can give you the answer, or basically we can provide you the solution or the whatever that you're seeking, your expected results in 5 days time. Okay, so how important it is when you use M13 tail primer. So it greatly increases the short sequence quality, because now the primers that we are using is no more the primer that you are using for your amplification purposes, but we are using the M13 tail, or basically M13 primer, to sequence. So now you get a full coverage of your sequence, okay? And then though you are using degenerate primers in your amplification, now you do not need to worry, because now we have a more specific M13 primer that can be used for sequencing. So it's more accurate. It is also less error. Okay, and of course the final one you do not need to clone, so you can just send us the qPCR product and we will do the rest for you. Now let's look into what is our workflow, basically the workflow from our lab. Now you know that WHO, okay, WHO also has their own protocol. CDC also has their own protocol, and basically the genes that they use to screen for COVID-19 is different. So WHO actually uses RDRP and E gene. Okay, so this is the list. So we have the forward, the reverse primers for both. And for CDC, we have N1 and N2, the primers and the primers for N1 and the primers for N2. Okay, so what we are going to do is, this is the M13 tail sequence, which is about 17 base pair in length. Okay, so we will insert this particular primer, which is you see the forward, you see the reverse, at 5' of the primer. So for example, if you are working with RDRP here, all right, so this is the forward. So this is actually the primer that is used to amplify. So what we'll be doing is we will add in the M13 primers. So these are the list of M13 primers, or basically we have all these ready in our lab. Okay, these are the primers we have. So basically, if you're using these primers, all right, you can just let us know, and we are on the go to help you to work out this particular project for you. All right, so what happens is, first, of course, after finishing your qPCR, okay, you submit the samples to us. So what we will do is, basically we will do a PCR. So in this particular PCR, we will be using M13 tail primer, like just how I explained earlier, all right, to amplify back the product that you have. And then we will do a PCR cleanup, and then we'll subject it to Sanger sequencing using M13 primers. And finally, we will submit the results to you we will submit the results to you. And there are few things that we also will submit. So one of it is codon aligner, where we will tabulate the trace view, the mutation, and also the raw data, the sequencing raw data that is also will be sent to you. Now, for your information, the first time when you send us your samples, so all optimization, because the first time you are sending, so when you are sending it for the first time, so we need to synthesize this particular primer. So which means we will still add these particular M13 tail primers to this particular primer, whichever primers that you are using, okay, be it NE, or even if you have any other genes, okay, so we will add M13 tail into it. So it actually takes approximately about 2 weeks. So this is inclusive because we have to also order and synthesize the M13 tail with the primer sequence of what you have amplified in your qPCR product. So this is only for the first time, but the subsequent order, the turnaround time will be about 5 working days. So what do we do during that 5 particular days is definitely upon sending the samples, we will actually quantify the DNA sample, and then we will do M13 tail PCR amplification. Okay, and following that, we will do a purification, we will do a forward and reverse Sanger sequencing, as well as alignment of with the reference. So we'll have an alignment of the reference gene, which actually perhaps the customer or you will provide us. So we will align back with E gene, RDRP, N1, N2, and then we will do a codon analyzing, and we submit the results to you. So what is happening before is this is the gene. So basically, before even M13 tail primer is actually inserted, so you can see basically the gene size is very small. That is about 100 base pair, 113, and N1, N2 is less than 100 base pair. So when we increase, when we insert the M13, so now it goes approximately about 100 plus. So all are sitting at 100 plus. So it's now much more easier to sequence, and you get a full coverage of these particular sequences. So what are you ought to expect after this particular sequencing? So overall results, what you're going to get is you're going to get quantification data, which means once you send us the qPCR product, okay, so we will do the PCR, and then we will do a quantification, which means after the M13, we do a quantification of the PCR product that is being amplified. Okay, and then we will also give you the raw sequencing data, which is the chromatogram. So you get for both forward and your reverse. So the number of samples that you send, so everything we will have the forward and the reverse. And then we will also give you alignment results from the codon aligner. So here you'll have the contig view, trace view, and the tabulation of mutation. So if you want to see, so this is the raw data. This is example of RDRP M13 reverse. So this is what you will also get. And this is from the codon analyzer, where this is the contig view. So if you see, this is the reference. So this is all the sequences that we have obtained, that they have amplified from the samples that you send. So what we do is we will align back this to the reference. So now you actually can see where's the mutation, and for maybe for your downstream works or for your downstream studies. Okay. And this is the trace view, because we will also align forward and also the reverse. So basically you get to see the alignment of all the samples. And finally, the mutation tabulation. So this one perhaps will let you know how is it different compared to the reference. So it will tell you where's the start, where's the end, and then where is the mutation found. So you can see the content here. So this is the content that you can actually look into the mutation. Okay, so how are you going to prepare samples and how are you going to submit? So basically, there are a few information we need when you order this particular service with us. So basically what we need is we need a primer sequence information. Why? Because you would have used primers to amplify your qPCR. So these particular primers are the primers that we will be using, basically the sequence we will be using to insert or add M13 sequences inside. So basically we have to resynthesize the M13 with the primers. So that is why this particular sequence is needed. So if in case you are working with like all these COVID-19, you are screening for RDRP, E gene or N gene, it'll be also good that you let us know so that we can always cross check to make sure that the primer sequence that you're using is similar with us. So it's only a first time. So if there's any changes, then perhaps you can let us know. So the first time, maybe we need to know whether these primers are aligning or in line with whatever that we have. So if it yes, then we do not need to order these particular primers. Yeah. And then also to submit the reference sequence. So for example, now you have your N gene, S gene, or whatever genes that you want to amplify. So all these genes should also cover the priming site, which means the primer that binds and the region. So the target region has to be there. And also it's we want to also let you all know that to submit unpurified RT-qPCR only. So basically, for you all just need to submit unpurified qPCR products only. And don't forget to fill up the biohazard form. There's also an attachment in the form where you can actually fill the biohazard form. So this is the sample submission form. So basically like how you have submitted for Sanger sequencing. So you just have to fill up your name, your address, so which is the one part above this project outline. So once you come to project outline, so you get to choose whether you want to screen for COVID-19, all right, or you have other genes you want to so it's not only for COVID. This presentation is for COVID, but you can also send in for other genes, perhaps dengue, if you want to look into some dengue genes, or any custom based 13 tail sequencing. So we can also customize it for you. So these are some of the projects that we can actually take in. And then you can name the gene. This is the primers that we hope to get some information with, so that we can generate the M13 sequence and reference sequence. So you can actually let us know which samples you can, what type of samples, basically qPCR or PCR products. So this is also another biosafety, which is the biohazard form. So you can click them to write this, to fill up the form. Okay, so basically all these particular form, when you're interested in into our services, we will send this to you. Okay, and of course the number of you can send in the templates. So basically, what is the name, number of templates, any remarks that you want us to look into, you can always write them here. So basically the conclusion is M13 Tail Primer amplified products gives a better sequencing result. So literally why I'm saying this, because you do not need to be afraid if you are losing one part or another. So you tend to have a full coverage of the sequences that you want to look into. And then the workflow is literally simple, because it is you just have to send in the qPCR product. There's no other work here. Just prepare your qPCR product, fill up the form, and we will do the rest for you, including tabulating or perhaps looking into the sequences, where's the mutation located. So all this we are actually will be providing to you. And then like what I said, you can actually use all these particular sequences for other analyses. Maybe you want to find some community identification or mutation screening or any other downstream project or objective that you want to achieve. Now, this M13 is definitely an excellent choice to replace cloning and transformation, because like what I've explained earlier, cloning transformation takes time. It's much more longer. It actually, when you maybe cloning and transformation takes a week, and then you send it for sequencing, analyzing. So all this may take time and also cost. But now with M13 Tail Primer, now you can just it's much more faster. Literally, you can just finish it within 5 days, and you have all the information that you need in your hand for your other downstream work. So that's all my sharing today on M13 Tail Primer. Thank you